Good morning, ladies and gentlemen. Welcome to day 2 of Canola Week. I'm Warren Ward, an agronomy specialist with the Canola Council of Canada, and I'm our team's lead on nutrient management. And coincidentally, that's the theme of our Canola Discovery Forum today. The Discovery Forum and the rest of this conference would not be possible without the support from our generous sponsors. A huge thanks goes out to all of the sponsors of Canola Week 2024, and they're listed on the back of your programs. For those of you who couldn't join us yesterday, I'd like to remind everyone to use the mics when asking and answering questions. And this is to ensure that our online attendees can hear your questions and answers. Each session has 2 mic runners, so if you have a question during the question and answer portion, just please raise your hand and we'll get a mic over to you. In addition, you can use Slido to type out your questions, and there's access to Slido using the QR code, which is on your name tags as well. Or if you're joining online, you can use it there as well. So with Slido, just remember if you see questions on there that you're interested in as well, you can always upvote those questions, and you can do that by clicking on the thumbs up icon, which is located beside the questions. And this will just help our moderator select the most popular questions for that question and answer period. Any of the virtual attendees who are having technical issues, please send a message in the Zoom chat function, and our team will give you some assistance there. For those collecting CCA credits, we'll show the QR codes on the screen at the end of the day. For those of you in person, you can also sign in at the registration desk. Now I'd like to take this opportunity to recognize that Saskatoon is in Treaty 6 territory, which covers most of central Saskatchewan and central Alberta. First Nations on this land include Cree, Saulteaux, Blackfoot, Dene, and Nakota. This is also the homeland of the Métis. It has been, for centuries, a place of exploration and discovery. We can think about that today as we're participating in the Discovery Forum. We used to host Discovery Forum as a standalone event, and the goal of the day really was to foster an environment of knowledge transfer, explore ideas, and help shape messaging and research priorities for the industry. I've been our agronomy team lead for nutrient management for a number of years now, and when I first took this on, it was a priority, but it really has become more so in recent years, with an increased focus on research projects and emissions reductions. As you know, many of these research projects that are currently ongoing or completed can be found on the Canola Council website under the Research Hub. The science edition of Canola Digest is also a good place for reading up and getting a take on some of these research projects. And you can also find some of that information in some of our topics on Canola Watch that comes out weekly during the growing season and once a month here throughout the winter. The federal target to reduce emissions has only made nutrient management more important to our industry. And as we remain proactively looking to improve canola production, that's a big focus for us. Another major focus for us has been on 4R nutrient management. And that's using the right source of fertilizer at the right rate, at the right time, and in the right place. Within that framework, we've had agronomic campaigns to increase the awareness of 4R management in an effort to help increase the adoption of these 4R practices. This includes things like the importance of soil testing, which has traditionally been a limiting factor for 4R adoption for many growers. Another focus for the industry has been to set appropriate yield targets. Now that would be realistic yield targets, but not limiting yield targets, so we want to make sure we're capitalizing on our production capabilities. Also moving from managing fertilizer from a farm level to the individual fields and then to zones within the field. Over the last few years, many new agronomists have obtained their 4R designation, and this shows the commitment to this process in assisting producers in making 4R a part of their management plans. Terms like nitrogen use efficiency are being taken from research and put into practice as we recognize the need to support the adoption of practices and products that drive improvements in our fertilizer management. These management decisions not only benefit the environment, but they also must make sense logistically and financially at the farm level in order for producers to adopt them. Our goal is to continue to increase canola yields while balancing that with our industry's sustainability goals. I would say that in general, we do a good job of producing canola, but we can always look for opportunities to improve. And again, that's another theme of the Discovery Forum, is that that knowledge sharing, the ideas, and really looking at what next can we do. Looking around the room at the questions that came in yesterday, I would say that the knowledge and experience in attendance at Canola Week is immense. The information and topics represented today come not only from our excellent lineup of presenters, but also from all of you that are in attendance. With that in mind, I'd encourage you to ask questions, give suggestions, and keep the conversation going during the breaks. Just don't forget to come back after the breaks so you can hear the next presenter. And this evening, wherever your plans take you as well, just keep that environment of learning and discussing ongoing. The last time we focused on nutrient management at Canola Discovery Forum, I tried to make a joke about nitrous oxide and reference the Fast and the Furious, but I think only about 3 people got it. So I'll spare you my stand-up comedy act, and we'll get right to our presenters today. So our first session today covers nutrient management research updates, and we're going to have 3 speakers, and after each speaker we'll have a question and answer session, if time allows. We had lots of great questions from the audience yesterday, so please continue to engage today. Now, first up, our first presenter is Reynald Lemke, and he is a research scientist and team lead on agronomy and environment with Agriculture and Agri-Food Canada here in Saskatoon. He will address the question, does fall-applied nitrogen influence nitrogen loss during the overwinter and spring thaw period? So, Reynald, I'll welcome you to the stage, please. Clicker's right on the table.
Well, good morning, everyone. I'll start by thanking the organizers for the invitation to come and talk. And let's see if I can figure out there we go. I'm going to jump right in with acknowledgements at the start so that I don't get excited and forget all about this, run out of time at the end. So I definitely want to acknowledge the funding support that came through the Canola Agronomic research program and the in-kind support from Ag Canada. And then a huge thanks to the technical staff that worked on this project. Crazy scientists got this idea of looking at emissions and N transformations over the winter period, but somebody actually has to go out there and take the measurements and deal with the inclement weather and et cetera. And so they did a bang-up job, and I definitely want to give a shout-out to them. So this session is about nitrogen use efficiency, which is really all about getting as much of the applied fertilizer nitrogen into the crop as possible. I'm going to talk about sort of the opposite end of that and looking at potential losses, and particularly in the overwinter and spring thaw period. So when we look at nitrogen fertilizer applied, the opportunity and pathways for loss are many. Most of the fertilizer that comes that is applied is either as a urea or ammonium-based form, although you may get some small amount in the nitrate form. And of course, most of that is nitrified fairly quickly over to nitrate, and so you have some opportunities there for loss there, ammonia volatilization, N2O and N oxides. But when things really happen is where you get wet conditions or reasonably wet conditions and available nitrate. You have lots of opportunity for denitrification straight up, which is moving that nitrate to N2 or N2O, or nitrifier denitrification, and then of course there could be leaching, surface runoff, and so forth. Each of those, just for completeness, whether it's ammonia volatilization, leaching, or runoff, also has the possibility of secondary emissions of N2O or N2. I'm going to focus, there we go, really for the next, for the balance of this talk, really on this area of denitrification. That is moving, as I said, N2O to N2. We can measure the N2O, and we're interested in that because it is a powerful greenhouse gas. It also interacts and catalyzes destruction of the ozone layer. But remembering that generally, when you see N2O emissions, it's a signal that there's probably wider N loss, sometimes much, much higher losses of N2 than N2O, but we can't, unless we have very sophisticated approaches and technology to do so, we cannot measure the N2. So we can only measure the N2O. So as I said, things really start to happen when you have the confluence of moisture and available, particularly nitrate. So for the prairie region in particular, generally what we see is a typical sort of bimodal seasonal emission pattern. So emissions happening there on the left of the screen, which is after seeding, fertilizing, assuming it's a one-pass seeding operation, and you may get emissions depending on rainfall patterns and et cetera. We've known for a long time that the snowmelt period and spring thaw is a high emissions event, but it's a complicated area to look at. Some years we get very few emissions, other years we get extremely high emissions. We've also known for a long time that nitrate, applied in the fall tends to disappear by the next spring. Research coming out of the 70s and 80s, sort of using N15 and so forth, have demonstrated that very clearly. But for here on the prairies, we've always assumed that if we apply urea or ammonium-based fertilizers very close to freeze-up, when the soils are already cool, that during our long, cold winters, soils are deeply frozen and not much is going to happen. Our colleagues in Eastern Canada have shown that there are emissions there over the winter period, and they can be significant. But sort of from our perspective, you're saying, well, they have, deep snowpacks, keeps the soils, a lot warmer than here. Maybe surface soils are near the thaw point or not too far below. And, we think that not too much is happening. But it's always been this niggling question as to whether that was actually accurate or not. There has been a recent publication out. Colleagues at the University of Saskatchewan, Rich Farrell, Kate Congries, and in association with Claudia Wagner-Reynald, who's here today, using the Tower flux approach, measured emissions through the winter period, through the spring thaw, and did in fact show that there are sort of ongoing low-level emissions. Now, in this particular project, what I proposed to do was deploy a automated flux or semi-continuous, I call it, and I'll show you that in a moment, flux measurement system. And that allowed us to be able to look at treatments. So in this case, we wanted to quantify soil-emitted N2O from soil receiving urea or a dual inhibitor, nitrification urease inhibitor, where Super U is the trade name product, and compare that to a no-N fertilizer. And then see if we can dig down into, assuming that we're going to see emissions and so forth, what are the factors that are involved in driving that, and then finally to dig even a little deeper and look at soil microbial dynamics and so on. Won't have time to talk too much about that final. So the approach we took, I'll try and go this really quickly, is we set up a field site at our research farm off Low Road here in Saskatoon. It was, we picked a canola stubble field, set up small plots, and then, as I said, laid out 3 treatments, a check, 100 kilogram of urea, and 100 kilogram of Super U. Treatments were applied October 31st in 2022, October 17th. We were trying to get as close as we could to kind of the onset of winter. In fact, October 31st in 2022, we ended up with a huge snowstorm the day after, or actually as we were wrapping up, so that was about as close as one could get. And we deployed, as I said, an automated semi-continuous measurements. In other words, we would get from every sampling position, we'd be able to measure that within a 24-hour period about 4 or 5 times, and we could measure N2O, carbon dioxide, and methane. We also used PRS probes to try and monitor N transformations, other soil samples both for nutrient transformations as well as the microbial work. That's the system there. You can see the chambers, hopefully. This was a snap that was taken on January 9th of this past January. There's a trailer set up there where our analytical equipment are included. So each of those chambers collect up with tubing and electrical lines into the chamber into the trailer where we have FTR taking measurements. So the way this runs, one of the chambers will close. In this case, we had it set for about 20 minutes runtime. It'll draw a sample in through the analytical equipment and return it to the chamber. We'll get a measurement over that time period. It opens, moves to the next chamber position, which then closes, and you work your way around. So that way, as I said, depending on how many chambers and how long you have the runtime and so on, will depend on how many times you can measure in a 24-hour period. In this case, it was about 4 or 5 times every day. And we tried to keep it running 24/7, but as you can imagine, these are challenging conditions. The system wasn't really designed for overwinter, but it did surprisingly well, actually. But much of that is to compliment, I guess, my technicians' over and above efforts to keep things going. So what did we see? I'm going to start with CO2 emissions, and looking at CO2 really was an indication, I guess, in this case, of biological activity, most likely microbial activity, and I've just sort of accumulated the emissions rather than showing you sort of daily fluxes over a 2-month period, just to give you a sense of kind of what we're looking at and what we were measuring. So in 2022-23, during November, December, this is CO2 kilograms of carbon per hectare. And so you can see that November, December, the cumulative through those 2 months was, I don't know, around the 100 kilogram mark or something like that. So short story is we were seeing emissions throughout the entire winter period. They're low. So during the summer, we're probably seeing perhaps, several times that amount, maybe order of magnitude. But in any case, what this demonstrated, I guess, is that there were indeed emissions, which suggest there's microbial activity, which would suggest that there's N transformations happening. So you can see the totals there, just over maybe about half a ton or so of CO2 carbon through that entire time period, November through to the end of April. In 23-24, much the same. In this case, the emissions were a little bit lower, but it still suggested there's sort of ongoing activity even through the winter period, January, February. What you don't see here, because I've sort of accumulated this across a couple of months, is that by early March, the CO2 emissions had pretty much shut down. Now, we don't know for sure if that meant that the activities had stopped or if, in fact, the soil surface had frozen over so well and capped off, and so it wasn't allowing exchange with the atmosphere. But in any case, all indications are that we do have activity through that entire time period. And as I said, suggesting that we also have N transformations. So this is looking at, and I struggled with how to best present this so it wasn't misleading. I'm not sure if I managed that or not, but I'll try and explain what I'm showing here. These are the PRS probe, plant root simulator probes, which are ion specific, and so they accumulate either ammonium or nitrate. They're separate probes for each. And we installed them immediately after the fertilizer was applied in the fall. I did one exchange over a 4-day period. That's what the 4-day period, 160-day period, and so on is about. So over that 4-day period, this is what accumulated on the probes. Now the units here are, if you're not used to working with these, are kind of funky. It's micrograms per 10 square meters per period. Essentially, it's a relative comparison. I mean, if you're doing this during the growing season, you can relate this to sort of plant availability and come up with fertilizer recommendations and so on. But what we're using it here is really just a relative comparison. First of all, are we seeing some accumulations of nitrate and ammonium? And also, is there a difference whether it's on a urea treatment or a super-urea treatment or on the control? So you can see in that first period there, in the first 4 days, very low. There was a little bit of both nitrate and ammonium showing up. Then the soils froze. We left the probes in place through the entire winter period. Again, they weren't really designed for that necessarily, but we thought we'll give it a shot and see how it goes. We pulled them out on April 11th, and you can see that a fairly large amount of particularly nitrate, that's in the blue bars, had accumulated, and a certain amount of ammonium, but much less. There were some differences there. I don't have the stats showing here, but the middle bar is urea, or the middle pair of bars, I guess, and what we're finding is that on the urea treatment, nitrate was higher, but not a lot higher than the control, and on the Super U, it was also higher than the control, but not as high as the urea, suggesting that the Super U was doing at least slowing down the process of transformation during that overwinter time period, but there was more happening than on the control, and there was even more happening on the urea. So then around the 13th or so of April, when things were really starting to thaw out and things, snow was gone, etc., we did another exchange, and you can see there, of course, the numbers are much lower because it's only a 6-day accumulation period. And again, we're seeing that urea had higher nitrate and in this case higher ammonium compared to the Super U and certainly compared to the control. And I see my red light is flashing, so I'm going to hustle along here. And then finally, at the kind of the end of April, similar but even more of a difference on the urea treatment. So very quickly then, did this translate into N2O emissions? And yes, it did. We were seeing emissions. Again, we've accumulated this across 2-month periods. It's grams of N per hectare, small amounts, although there's a good chance that much larger amounts were accompanied with this as N2. In any case, we were seeing emissions during November, December, not much difference in treatment. Same thing through January, February, and then finally kind of through the spring period, March, April. Not a lot of activity in this particular year, and the presence of either urea or Super U did tend to increase emissions. And in total, you can see that urea was slightly higher than Super U, but not statistically different. Pretty much the same story for 23-24, except here the differences were a little higher on the urea. We were seeing higher emissions than, say, on the Super U, and certainly higher than the control. So the short story, I guess, there is that it does, in fact, show that there are N transformations that are ongoing through the winter period, even when you get that fertilizer in very close to freeze up. And although the emissions are fairly low as far as N2O, we don't know for sure, but there's a good chance they're accompanied by much higher emissions of N2. I'm going to skip over these last two. Just a quick comment here. We're finding some rather interesting results in the microbial where we're seeing some differences in the NOSI or the clade 2 as NOSI, which maybe doesn't mean a whole lot to many of you, so not going to talk about it, but it's just kind of a teaser that there was certainly activity amongst based on the gene numbers that we were measuring over the winter period, and there sort of hints at the possibility for that there might be other ways that Super U might be reducing emissions as opposed to just slowing down nitrification rates. So quick summary then, soil biological activity did continue through the winter period, as did nitrogen transformations, as did N2O. Cumulative losses were low, but the fall applied fertilizer did increase N2O emissions even when we got very, very close to freeze-up, and the dual inhibitor reduced but did not eliminate that increased risk of N loss during the overwinter and spring thaw period. And I think I've run over a little bit, but hopefully not too bad.
Thank you, Reynald. Yeah, and we do have some time for questions. I see there were a few coming in online that we can get to, but also if you have questions in the room, please raise your hand and we'll get a mic over to you. Maybe just to kick things off, a quick question about the site you were using. Do you know what the background levels of nitrogen were on the site? Did you target a high or a low background level of nitrogen in the field?
Yeah, I do, and I have the numbers, but I don't have them here, but we were targeting low. So the year, sort of for the setup for this, we grew canola. We applied a fairly low rate of nitrogen to try and ensure that the nitrate levels in the fall were fairly low. But it is it's a pretty nice soil. It's a clay loam, so just the natural levels, I guess, ambient levels for nitrate are, I mean, you can certainly pick it up in the fall.
Yeah, we have some online questions here. So how deep was the fertilizer placed in this study?
We banded it in the fall as deep as we could try and get it. We were dealing with small plots. We had a small plot bander, so we were shooting for about 3 inches. I'm not sure if we quite got it, but that was our target. So it was definitely banded. And we tried to do the best job we could of getting to depth and getting good furrow closure.
Okay, we've got a few more.
Are you planning this to be a longer study? Because last year was a very different year, talking about winter in Saskatchewan.
Actually, this study has wrapped up officially, and it's a good point. It turned out that 22, 23, 24 were very, very different winters. So I think we got a fairly good range of conditions. But, I mean, I don't know, what is normal these days in terms of, you know, so we only will have, well, officially we're funded for 2 years. We're going to try to make the attempt to run it for a 3rd year if we can pull it off. But that would be the best we have.
Okay. How much of a factor is soil moisture and rates of nitrogen transformation?
A lot. Yeah, not sure how to quantify that answer, but certainly temperature and moisture have a very, very strong impact on. I guess the temperature question, though, is, I mean, obviously during sort of growing season, things are going to happen much, much faster. And it was one of the reasons that sort of the overwinter period, we thought, well, maybe things aren't really going on because temperatures are quite low. So I guess what we're seeing here is suggesting that they do continue even under very cold conditions, although it's very slow relative to spring.
We got a few more here, Warren. Good time? Yeah. Okay. Hey, these 2 seasons had very different snow cover conditions. Did you also look at soil temperature feedbacks, and is that part of the story in describing this?
We haven't really had a good chance to, so yes, absolutely it is. And of course, snow cover will insulate and So there's sort of an interaction here, I guess, with do temperatures drop well below freezing before we get a lot of snow or vice versa? And it's going to make a difference on sort of how fast the soil profile freezes up and so on. We do have soil temperature data, haven't really had time to really dig into that and work with it, but definitely that's the next step that we need to go at. But the short answer, I guess, is that that interaction between snowpack and the timing that it comes and the soil temperature through the time period and how fast it freezes, how deep it freezes, clearly there's a strong interaction there.
What was it? How does 300 to 400 grams per hectare compare to what would be emitted in May, June, or when the soil isn't frozen?
Depends on the year, but you know, I guess kind of a rule of thumb would probably for a site like what we were working on, we might be looking at 500 to 1,000 grams lost during growing season. Huge variability depending on year and timing of rainfall and so on. So it could be as much as a 1-to-1 or, you know, maybe 50% of the total emissions, something like that. So if we're looking at 400 grams. So a small amount, but remembering that N2O is a very powerful greenhouse gas, so it's, you know, close to 300 times more powerful than CO2. So even those small amounts are significant from an atmospheric point of view. If they were accompanied by N2O emissions, which might be 10 times or greater what we're measuring, then the actual amount of N loss could be significant in an agronomic sense as well.
Hi, Reynald.
Morning.
Just putting it into a farmer perspective, say if you applied 100 pounds per acre, 100 kilograms per hectare of urea in the fall, or anhydrous ammonia, which tends to be a common. Mm-hmm. If you have a wet spring, lots of standing water, like exceeding the soil capacity, how much of that 100 is going to be left?
That's the million-dollar question. There's certainly opportunity for, especially in the low spots and so on, for significant denitrification loss. And I guess ultimately what we were sort of going after here is if there is transformation during the winter period, so now you've got a certain accumulation of nitrate, is highly susceptible to loss. And you know, from previous studies, nitrate, you could lose 50% and beyond of nitrate that's there. Now, in the spring, of course, nitrification starts to kick in as well. So if you have a low spot that's fairly saturated for a fair time, the soil's already warming up, things are really happening. So there's a good chance of high loss, and you could have, you know, 20, 30, even up to 50% of that. If it's an ammonium base, it's probably going to be lower, but I'm throwing darts now. So I think the best you can say is there's a potential for a fairly significant loss.
Please join me in thanking Reynald again. That was excellent. Good questions as well. Next up, I'd like to call on Rajeev Dhakal as our next speaker. Rajeev is working to finish his PhD on canola nitrogen use efficiency with the University of Saskatchewan and Agriculture and Agri-Food Canada here in Saskatoon. He also recently joined SARDA Ag Research as a research scientist just last month, and Rajeev's presentation will be titled Nitrogen Use Efficiency in Spring Canola. So Rajeev, welcome.
Need to learn how to use this first. Good morning, everyone. Thank you, Ryan, for a nice introduction. Today I'm going to talk about nitrogen use efficiency in spring canola. Setting up a little bit of background, canola is a very important crop in Canada, and its cultivation is mostly concentrated in three prairies provinces. And Canola Council had a mission to increase the yield up to 52 bushels per acre. And with 5 different tools, and there are 2 different tools that is of my interest, fertility management and genetic improvement. Talking about the nitrogen fertilizer applications, it's a report from 2018-19. Over 2.6 million metric ton of nitrogen is applied in Canada as a fertilizer, and majority of it, it's more than 35% of it is applied in canola. And with the recommendation rate going higher with the recent research publications, recommending up to 165 kg per hectare to have a very good yield in hybrid canola, the use of nitrogen is not going down. And we know that in the year of application of nitrogen fertilizer, the fertilizer nitrogen use efficiency is only 50%, meaning really exceeds 50%, meaning 50% of the applied nitrogen is left in the soil, either to be used by the crop next season or to be lost in the environment. And Reynald put out like different ways of loss. I don't want to go into detail about that. Talking about the research in fertilizer management, there has been a lot of research on how to manage nitrogen fertilizer for efficient use. Like 4R steroid is one of the prime example. But then what we see is there is a missing piece of puzzle, like we are yet to look into the genetic variances or differences in genotype. And that's where my research focuses more on. So to start this talk, there are 4 different questions that I want to set up and ask while doing my research. The first thing is, where do the nitrogen go within the plant during the growing season? And is there a variation for nitrogen use efficiency in spring canola? And what are the traits that are associated with nitrogen use efficiency in spring canola? And other thing we wanted to see was, has nitrogen use efficiency in spring canola has improved indirectly with the improvement of canola throughout the year? And we also wanted to see like what's next there regarding nitrogen use efficiency in diversity, canola diversity population. Before starting my results of my experiments, let us be familiarized with the nitrogen use efficiency matrices that I've used during my research. There are a lot of nitrogen use efficiency matrices that has been used by different researchers, but I decided to we chose like 5 of them. First one is yield-based nitrogen use efficiency. It says it represents the yield produced per available soil nitrogen. A second one is crop-based nitrogen use efficiency. It reflects how well the soil plant take up nitrogen and put up in yield, nitrogen in yield. A third one is nitrogen uptake efficiency, how it represents how well nitrogen is taken up by the plant and it places in their shoot biomass. A fourth is nitrogen utilization efficiency, like when the nitrogen comes to in plant, how well it uses to produce yield. And finally, nitrogen harvest index that represents nitrogen remobilization from shoot to seed. So the first experiment was carried out with 35 different historical cultivars. I mean historical because they represent 70 years of canola research and development in Canada. So there were 36 open pollinated varieties and 9 hybrids. And 26 open pollinated varieties and 9 hybrids. Among open pollinated varieties, there were release varieties and there were some breeding lines and some of the open pollinated genotypes that were used in breeding. And among hybrid, most of them were commercial and one was commercial, experimental hybrid. Research trial was carried out in 2019 and 2020 in Agriculture and Agri-Food Canada research field at Saskatoon and Melfort. And we used, like, those 35 different genotypes were grown in 2 different nitrogen levels, low and high. And we sampled the plants at 4 different growth stages for tissue analysis and biomass. So first question, where does the nitrogen go? Like, if we see here, during vegetative stage, most of the nitrogen and most of the biomass, proportionately, it's in leaf, and the leaf biomass and the leaf nitrogen content gradually decreases towards as the crop develops. And the major differences lies during the maturity stage. The proportionately canola plant more or less have similar amount of biomass distributed across the different plant parts, seeds, stem, and cilic husk or pod walls. But if you look at the nitrogen, more than 60% of nitrogen is in the seed. So, and other difference between biomass and nitrogen content is like more than 90% of nitrogen is acquired by the plant by the time it flowers, but the biomass peaks by the time it's maturity. So let's see whether there are differences in hybrid cultivars and open pollinated cultivars or not. If you look at the low nitrogen treatment, we didn't see much of a differences in terms of actual total nitrogen content in plant. They're almost same. But if you look at the high nitrogen treatment and look at the maturity stage, hybrid cultivars tends to put more nitrogen from soil to the plant, in plant. And then regardless of the nitrogen treatment, proportionately, hybrid cultivars tend to push more in-plant nitrogen or proportionately distribute more nitrogen towards the seed than open pollinated cultivars. So they are efficient in utilizing the in-plant nitrogen once it's in the shoot. And the other thing we were looking was variation, whether there is a variation in nitrogen use efficiency traits. We saw that nitrogen use efficiency matrices like yield-based nitrogen use efficiency, nitrogen uptake efficiency, and nitrogen crop-based nitrogen use efficiency greatly varied due to the site years or the environment, while the variation due to genotypes were higher in nitrogen use efficiency matrices like nitrogen harvest index and nitrogen utilization efficiency. So what are the traits related to nitrogen use efficiency matrices? I'm sorry about the low, very small font, but the summary of the whole this graph is, yes, there are traits that are highly associated with nitrogen use efficiency matrices, and if you want to group them, here we are, like harvest index is highly associated with nitrogen utilization efficiency and nitrogen harvest index, and yield is highly associated with crop-based nitrogen use efficiency and yield-based nitrogen use efficiency. While shoot biomass and nitrogen content in shoot biomass is highly correlated or associated with nitrogen uptake efficiency. Moving on, the fourth question, has nitrogen use efficiency improved or changed over the years? So if you, we can see with this graph, this is the graph that I prepared from Stat Canada data. That we can see the yield has increased since the first variety of rapeseed was registered, and coming to 2023, yield has increased. But then we can see the two distinct slope. Then we're curious about, is there a broken line relationship? And if we can find the year where the yield has started to accelerate in the higher rate, and we carried out a segmented regression. So basically, it separates the broken linear regression in broken lines form, and we found out year 2001 was the breakpoint that happened to match with the point that Greg made yesterday. So that was a kind of a stimuli for today's talk. So thank you. So then what we did was we separated data set into three different sets, like period 1, pre-2001, period 2, post-2002, and overall period, and carried out a separate linear regression, like for overall period and pre-2001 and post-2001, and tried to see how is nitrogen use efficiency matrices has changed over the years. So here's the first three graphs. The one on the left-hand side is for yield. As you can see, the varieties, the other cultivars that we have in our experiment follow the trend of Stat Canada production data. Like there is a higher rate of gain towards the after post 2001, and similar trend is followed by yield-based nitrogen use efficiency and crop-based nitrogen use efficiency. When you look at the nitrogen utilization efficiency and nitrogen harvest index, it shows improvement over the period, and the pattern it follows is of that of harvest index. The interesting point we found out was there was no such pattern of improvement in nitrogen uptake efficiency, meaning the nitrogen uptake efficiency remained almost stagnant through the 70 years, mainly because there is a stagnation of nitrogen in biomass, and there is stagnation of biomass. So other thing we wanted to do was like, we wanted to see more of the diverse spring canola population and its nitrogen use efficiency. So we thought like, how about taking Saskatchewan spring canola or Brassica napus nested association mapping population and population parental lines and see how the parental lines are differed for different nitrogen use efficiency. And I think there is a talk tomorrow who will be talking more about the nested association mapping population and everything. For now, we just need to understand that nested association mapping population is a very powerful tool to dissect the genetics behind the different complex traits. So it is composed of 50 founder lines crossed with one reference lines, and then there are more than 2,500 reels. Reels means recombinant inbred lines in the population. But I'm not using whole set of population, I'm just using the parental lines from that population. So this is experiment 2, and field trials were conducted in AFC farm at Saskatoon for 2019 and 2020. And same, like we had 2 different levels of nitrogen, and plants were sampled at different growth stages. And what I did was I tried to see whether there is a distinct pattern among the cultivars for different traits or not. And I carried out PCA, principal component analysis, and cluster analysis using 15 different traits, including phenological traits and yield-related traits, and those 5 NUE matrices. And from the cluster analysis, we've found out that more than 90% of variation is explained by 5 cluster. And from the figure from right, we can see that there is similar kind of association among the traits that we saw in the previous historical panel. And moving on to summary of cluster analysis, we see that genotypes in cluster 3 have higher nitrogen uptake efficiency, higher yield-based nitrogen use efficiency, and crop-based nitrogen use efficiency. While genotypes in cluster 5 have greater nitrogen utilization efficiency, nitrogen harvest index, and yield-based nitrogen use efficiency. But there was a distinct grouping at high nitrogen cluster analysis at high nitrogen, where cluster 4 had better nitrogen utilization efficiency. However, what we observed was there was no specific pattern of clustering based on the genotype, country of origin, or source of the genotype. So the take-home message, like long story short, is biomass, in-plant nitrogen, and yield are higher at high nitrogen condition, but nitrogen use efficiency matrices were higher at low nitrogen condition. Plant take around 90% of their nitrogen canola put in their body, like plant by the time it flowers. And among any different nitrogen use matrices, CITES differences affected more for yield-based nitrogen use efficiency and crop-based nitrogen use efficiency, including nitrogen uptake efficiency. But the cultivar variation were higher for nitrogen utilization efficiency, nitrogen harvest index, and harvest index. So looking at the trait associated with different nitrogen use efficiency matrices, so many nitrogen use efficiency. Yield is more related with yield-based NUE and crop-based NUE, and harvest index is more related with utilization efficiency and nitrogen harvest index. Similarly, biomass and nitrogen in biomass by the maturity is more related to nitrogen uptake efficiency. So overall, nitrogen use efficiency has improved indirectly, but nitrogen uptake efficiency didn't show any sign of improvement throughout the years. So if we want to look into the different diversity in future, so it's wise to look into genotypes at cluster 3 for nitrogen uptake, yield-based nitrogen use efficiency, and crop-based nitrogen use efficiency, and cluster 5 for nitrogen utilization efficiency and yield-based nitrogen use efficiency. With that, I'd like to thank my supervisor first, and I'd like to thank all the donors and all the people who made this research possible. Thank you.
Great, thank you, Rajeev. We do have time for a few questions, and I know earlier this morning Rajeev was mentioning his thesis defense is coming up, and he's really looking forward to a good question and answer session here, just as practice. So I see we have a few online. Is there any in the room before we get to the online ones? If not, I would just like to point out 2001 was kind of the year I first was really involved with canola. So I don't know what your thoughts are on causation and correlation and whatnot, but Sean. Oh, sorry, we have one at the back here first.
Just a question, like when considering like yield-based nitrogen use efficiency, was there differences between varieties and like the different nitrogen use efficiency?
Yeah, like there were differences between varieties that I didn't show that part, but yeah, there are differences between varieties too. Yeah, because it is mostly driven by yield. Like we applied same amount of nitrogen, like you'd expect a hybrid to be more efficient based on yield-based nitrogen use efficiency. And that's the thing when we decided to use more of the nitrogen use efficiency matrices, not only the way like yield produced per kg of nitrogen, because you'll see the hybrid would be the one that would be having more and will be satisfied with, well, we have improved the nitrogen use efficiency. But then if you look at the plant biomass and nitrogen in plant biomass, it's almost same. So it's just hybrid, they push more nitrogen to seed. They produce more seed, and when protein is multiplied by biomass, or nitrogen percentage is multiplied by biomass, that's the nitrogen content, right? So I hope I answered your question, yeah.
Okay. What factors do you think may have contributed to the increased yield, the increased rate of yield gain since 2001?
We covered that already.
Oh, okay. Yeah.
Him. No, I think it's a tremendous effort from all around. First thing is genetics. Second thing is agronomy. And we have done a lot of work in nitrogen management or fertilizer management side too. So those are major things. And pest disease, herbicide resistant, everything is there, and major was, everything came into a package of hybrid, right? So major was where we are transitioning from open pollinated varieties to hybrid with all those packages included. So that's the main driving factor, I guess.
Based on your research, what recommendations would you give a farmer?
Cool.
That practices would increase nitrogen use efficiency.
Yeah, like first thing is done by the notion that hybrids are better in overall have higher nitrogen use efficiency, but then be with cautious. And then for now, I think we have to focus on better nitrogen management that other researchers has suggested, because we are there to look into the genetics and slowly but surely we'll reach there, I guess. Yeah.
Okay, what plant characteristics or pathways do you think could be targeted to improve nitrogen uptake efficiency?
Oh, that's the key question I always ask myself. Like, you want to have better biomass, but you want to have better yield too. So I think we need to target harvest index as concurrently the total biomass. Like, not only target harvest index, but concurrently the total biomass. So that's the key.
Hey, what will be the approach to identify genes in specific clusters? Would this be a useful place to apply GMO or gene editing technology?
I think we are not there yet to use GMO or gene editing technology because we are yet to discover the genes. So from now, I think we need to target, like, look, what we did in the second part was we also sampled the reels. We have sampled the reels at different growth stages from those genotypes in those clusters. And then we want to do start with genome-wide association mapping, and then moving forward from there. I think that would be the pathway to go.
All right. Thank you again, Rajeev. That was excellent. Our next speaker in this session is Mervin St. Luce. Mervin is a research scientist at Agriculture and Agri-Food Canada in Swift Current and focuses on soil fertility and cropping systems. His presentation is nitrogen management and use efficiency in canola production systems. So welcome, Mervin.
Okay, good morning, everybody, and thanks to the organizers for inviting me to give a talk. Let me just make sure this is okay. Good. All right. So we all know that canola is very important in Canada as one of the most important crops, and canola requires a lot of nitrogen. Apart from water, especially in the prairies, where water is the most limiting for crop production, nitrogen is the most limiting nutrient for canola. And of course, it's required in the largest quantities compared to other nutrients. It's also applied, it's also very costly, as producers will let you know. As Reynald explained earlier, nitrogen is very susceptible to losses. It's very dynamic. So we need to be able to improve the use efficiency of nitrogen in canola production systems. Improving end-use efficiency, as the previous speaker has explained, there are many ways to look at calculating this, but in terms of nitrogen management, we need to focus on improving how we apply nitrogen, the rates, how we manage it in the field so that we can improve how much of that nitrogen applied is taken up by the crop. It is very challenging, but it is a necessary endeavor. We have heard of the 4R nutrient stewardship program to enhance end-use efficiency. That is applying nitrogen using the right source, the right rate, right time, and the right place. There are several factors, environmental, soil, other specific factors that can influence nitrogen use efficiency, nitrogen dynamics in the field. The focus of my talk will focus on two of those R's. So we'll focus on the right rate and the right time. So we did part of the previous cluster in 2018-2023, conducted a series of experiments with the objective to assess agronomic and economic responses of canola crop to end fertilizer management in terms of increasing end-use efficiency, grain yield, and standability. We also looked at improving end-use efficiency, crop productivity, and lodging resistance through best and management practices under different soil and climatic conditions. So those were just two of the, I think, 5 objectives that we had through that program. And we did have conducted 2 experiments. So the first experiment looked at the response of hybrids to end fertilizer rates and time. And the second experiment was a rotation study looking at canola frequency or intensity, and then fertilizer rates. I will not focus on experiment 1. We have published some of the findings, so I would direct you to this paper, and I'll be happy to talk to you about that after. So for this presentation, I'll just focus on experiment 2. So in experiment 2, looking at canola frequency and intensity and how that interacts with N fertilizer rates, this was conducted across 7 sites. So we had Beaver and Olds in Alberta, Swift Current, Melfort, and Scott in Saskatchewan. We had Brandon in Manitoba and Ottawa in Ontario. So we did have a wide variety of locations and conditions. The experiment, we had 2 main factors, the crop rotation, so the different intensity of canola. So we had an intense rotation of canola 3 out of 4 years. Then we had 2 out of 4 years and canola grown once in 4 years. Then we had our subplots as N fertilizer rates, 5 rates from 0 to 178 pounds of N per acre. So those were applied once canola was grown. And then those rates stayed into these plots unless the crop following was a pulse. So we looked at the sort of adding nitrogen in the same plot over a 4-year period once we started in 2018. So I would note that the experiment was not fully phased, meaning we only had 1 phase of the rotation present every year. Because of the size of the study, having every crop in every phase every year would require a lot of land to do that, so just 1 phase every year. So some of the field information, so we did the experiment under no-till conditions. The plot sizes varied by site, but generally about 6 meters by 15 meters. We seeded in May. We targeted about 80 plants per meter squared. That's about 7 pounds per acre, I think. Row spacing, normally about 12-inch. N was side banded, about 2 to 3 inches, so about 5 centimeters. Phosphorus, sulfur applied as needed based on soil tests. We did have some issues in the experiment. In Brandon in 2018, there was wet field conditions. We had clogging in the seeder in terms of the N applied. We had some errors in 2020 in Brandon and Ottawa, where we had some mix-up in terms of the plot plan, where the wrong plan was used, so the wrong crop went into the wrong plot, the wrong rate. And also, we had some challenge. We had in 2020 in Melfort, due to COVID restrictions, we couldn't actually run the experiment as planned, so just seeded wheat without fertilization in 2020. So this picture shows the differences you can see in Ottawa and Swift Current, which was very dry. Ottawa, we had rainfall, and you can see one side is green and the other side is brown. So that tells you the impact of weather conditions. Some of the initial soil characteristics, so we had differences in texture, soil organic matter content. You can see very high levels in Melfort and Olds, very low levels in Swift Current and Scott, differences in pH. We did target fields that had low N. However, we had quite high levels of nitrate in Olds and low levels in Scott. We also had differences in pH at the site, so we did try to get a variety of conditions to conduct this experiment. I put up this slide here, the results, just to show in terms of the organic matter levels and what we can expect in terms of the nitrogen supply potential of the soil, which is kind of related to organic matter content. So we did an incubation with the soils collected over 16 weeks, and you can see very high potential in Olds and Scott and very low levels in Swift Current and Scott. So just take that into consideration in terms of the response to nitrogen that will be applied in those sites. Of course, weather is going to have a big impact on the responses that we will find. We all know of the dry conditions we have experienced over the past couple years. So generally, across the 4-year period, we had mild to severe drought in most of the sites, especially in June and July, where rainfall timing is very important for canola. So we did have quite low rainfall. Similar for temperature, air temperature, we had above normal temperatures, especially in June, July, and August, especially in the prairie sites. So that did affect our results. So we had, in some cases, very low emergence. We had poor seed development, and that also affected during flowering stage in July. Okay, so I will go for each year one at a time. So in 2018, as you can see, we have differences among the sites. We only found response to N at 2 sites, in Beaverlodge and Melfort. We'll put an asterisk on the Brandon results because, as I mentioned, there were some challenges during seeding. So those are not really conclusive. Due to some difficulties at Swift Current, we actually seeded late, so that did affect our results. We seeded at the end of May in Swift Current. So we only found responses at Beaverlodge and Melfort. We have higher yields in Oles and lowest yields in Swift Current. And as you saw from the nitrogen supply potential, Oles has the highest ability to supply nitrogen. In 2019, so now we can look at the impact of not just the nitrogen rate, but look at the stubble, previous crop that was grown. So in terms of the rotation, so first you can see that we had a real yield response at 6 out of the 7 sites. So there was no response at Oles. We also can see that there are differences in terms of the yield potentials at the different sites. Now, in terms of the impact of the stubble, the previous crop, we did find a higher yield on wheat than canola stubble at 4 of 7 sites. So that occurred at Brandon, also at Oles, at Ottawa, and at Swift Current. Even in the other sites where the response was not, the differences were not significant, but you can also see that there was the same trend. Numerically, canola yield was higher on wheat stubble than on canola stubble. If we move on to 2021, again, based on the differences among sites, the yield varied across site. Yield response at 6 of 7 sites, so there was no response in Brandon. That may be due to possibly the errors that occurred in 2020. We had a negative response at Swift Current, which was quite interesting, and again, very dry in Swift Current in 2021. And now we can also look at the stubble, previous crop, as well as look at the intensity of canola over that 4-year period. So if we look at, if we combine the rotation that had P as the previous stubble versus wheat, we did find that we had higher yields on P than wheat stubble at 4 of 7 sites. So that's at Beaverlodge, Oles, Scott, and Swift Current. And then in terms of the canola frequency or intensity, you see yields were lower for the intense canola. So growing canola 3 out of 4 years, the yields in the 4th year was lower compared to growing canola once in 4 years. You see that the yields in Swift Current very low, almost non-existent, basically. Very dry conditions, and it's sort of the photograph I showed earlier in 2021. In Oles, it was also dry, so that's why the yields are very low. We had reports of poor seed development and seed pod shattering, so we had a lot of losses in Oles. So there's a little bit of asterisk we put on the results from Oles. Now, if you look at nitrogen use efficiency, and the previous speaker spoke a lot about that, in this case, we looked at, for this presentation specifically, just looking at the simplest way of presenting this, so just looking at yield versus nitrogen applied. So the first thing that you can see is that nitrogen use efficiency decreased as the nitrogen rate increased. That's the first thing that you can see across every year. So that tells you the importance of the nitrogen rate. Secondly, you can see that nitrogen use efficiency was higher on wheat than canola stubble at most of the sites, 6 of the 7 sites. And also that you can see that nitrogen use efficiency was higher on pea than wheat stubble at 4 of 7 sites. And also it was affected more by the nitrogen rate than the intensity of canola in the rotation. Before I go on to the next part of the presentation, looking at the economic optimum rate, I just wanted to explain how this is assessed. So if we look at a typical nitrogen response curve, and we want to find out the optimum yield, so that's the yield at which yield is maximized, the end rate at which yield is maximized. Beyond that end rate, there is no increase in yield. There is, most cases, reduction. So that would be how we would estimate the maximum yield potential. But if we take into consideration the price of the fertilizer and the grain that the producer is going to sell, and we look at economic optimum nitrogen rate, it will be lower generally. So now you can see that there is a larger gap between the nitrogen rate and the yield. So applying nitrogen above that economic threshold, there's no real gain, there is just additional cost. So before I present the next part, just wanted to explain that. So when we look at the economic optimum nitrogen rate, for some years we were not able to estimate that, and we only used one model for this presentation. Different models can be used, and we only used the quadratic model. So in 2019, what you can see in terms of the stubble, we can see that for most sites there's a difference in economic rate based on the stubble. And even with the same economic rate, there are differences in yield potential, economic yield. So we see in Beaver we have a little bit higher economic rate, but the yield is the same. If you go to Ottawa, we see a much higher yield on canola, much higher nitrogen rate on the canola stubble versus wheat, but we do find a higher yield for that lower rate for canola, for wheat as a stubble. In 2021, we also found that there are some differences in terms of the intensity of canola and as well as the previous stubble that was grown. You will also see that across the sites, the economic rates are different, and also the economic yield varies. Some years we were not able to be able to estimate due to drought conditions. Now, if we look at the site-specific economic optimum rate, so looking at all of the years, eliminating for very extreme conditions in terms of having values less than 0 or above 223 pounds of N per acre. So that's about 250 kilograms N per hectare. We see at Beaver Lodge, the economic rate is about 152 pounds with an economic yield of about 40. In Swift Current, it's about 75, economic yield about 29 bushels per acre. None of the sites have, in terms of these estimates, the target for the Canola Council, which is 52 bushels per acre. We did have 1 year where Beaver Lodge exceeded that. So we see differences among sites in terms of the economic rate and economic yield. So in summary, throughout this 4-year project, we found that canola response to N was highly dependent on the site conditions, especially due to precipitation and temperature, as well as nitrogen supply potential of the different sites. Importantly, the N application rate, so that's where soil testing comes into play, has a greater impact on the yield and nitrogen use efficiency than how many times canola is grown in a rotation. Yes, that will affect, because there is disease carryover and so on, that will affect the yield, but the nitrogen rate has a bigger impact, and of course that will be related to the timely rainfall and good growing conditions. We also found canola yield and nitrogen use efficiency were generally higher on wheat than canola stubble and higher on pea than wheat stubble. So the importance of diversifying rotations with legumes and different crops will improve not just the canola, but also the entire cropping system and sustainability on a whole. The economic rate was site-specific, but based on what we have found, so our recommendation is for about 80 to 130 pounds of N per acre for the black soil zone, 100 to 170 pounds per acre for the dark gray, that's in Beaver Lodge area, 90 to 130 for the dark brown, that's around Scott, and in Swift Current area, which is in the brown soil zone, about 75 to 100 pounds per acre, and for the Ottawa region, about 120. These rates also are in line with the paper that I put up earlier, where we looked at the economic rate based on different hybrids as well. So I would like to just thank the team involved in this project, all the collaborators, all the staff at the different sites, and of course all of the funding agencies for the kind support. So thank you.
Thank you, Mervin. We do have time for a couple of quick questions if there's any in the audience. I see there's an online one as well. Let's go online.
What was the cost of N and the value of canola used to establish EONR?
Very good question. So for this presentation, we just used, so across to price ratio, we used as 2, which we use in the paper, which is generally what has been used. So we did not specifically use the price of the fertilizer for this presentation. So if we estimate it at 55 kilograms, 55 cents, and then I can't remember exactly the cost of the fertilizer and the cost for the grain price that we use, but the ratio in terms of the cost and the grain price came up to about 2, so we just estimated it for the paper and this presentation as 2, so twice the amount.
Okay, thanks. Thanks, Marvin. Did you look at any kind of measure of mineralization throughout the season across the rotations, like with PRS probes or sampling?
No, so we did not look at N mineralization throughout the season in terms of PRS probes. That was part of the plan, but we were not able to do that. However, we did take soil samples at least once in the mid-season. We did measure biomass at least 4 times throughout the season. We had NDVI measurements at 3 sites. We looked at chlorophyll measurements at 3 sites throughout the season, but not PRS probes. So we can estimate nitrogen uptake throughout the growing season through the biomass. But I did not present this today.
Mervin, I was just wondering if you could clarify, you were showing your sort of recommended rates, the ranges. Is that application rate or is that available N? In other words, that to consider the soil N values?
Right, good question. So those are not considering the soil N, so those are total actual N application. So we are not, it does not consider the potential to supply N, does not consider the soil test. That's a total amount based on the responses that we found. It is important to do a soil test before the actual rate is decided upon, and then look at the yield potential, of course, to determine. But based on the conditions that we had and the results that we found, those rates seem reasonable for those sites, and they are actually in line with what is actually going on right now. So those are not new, actually.
ask them. Could a farmer plan nitrogen rates based on availability and amount of soil moisture?
Excuse me, could you repeat the question?
Could a farmer plan nitrogen rates based on availability and amount of soil moisture?
Yes. However, the soil moisture part is a question mark sometimes because we do not, that is soil moisture in spring, I guess, but we do not know what's going to happen throughout the season. So the general practice now is to look at soil tests generally in late fall, right before freezing, in terms of the timing and buying fertilizer and so on, compared to spring. So that's the general practice now, to look at just what is left in late fall and use that with the yield potential to estimate how much nitrogen is applied based on what is left. The moisture, that will improve the recommendation, but that is kind of a tricky part. There are different labs who incorporate this in the recommendation. Some also try to incorporate long-term weather forecasting, but that is, I would say in an ideal world, yes, but that is a bit of a, it's not across the board that we find. Throughout this project, we also want to incorporate this type of measurement in the recommendation. I did not present that, but we are working on the data set where we have moisture measured throughout the growing season. We will look at incorporating the nitrogen supply potential. So I've only showed the results of 1 year, but we did measure nitrogen supply potential through incubations throughout the 4-year period for every site, for every rotation, and see how the rotation and the nitrogen rates did influence how much nitrogen could be possible mineralized. So we want to incorporate that with weather and estimate how much nitrogen that the soils can supply. Most of the N, as you saw, in sites that did not respond, came from the soil, not from the fertilizer that was applied. So how can we take that into account? That is where the research gap is right now.
Great. Well, thanks again, Mervin. We'll stay tuned for those future results. So that wraps up our first session. Next up, we have our break. I would just like to take this opportunity to thank our gold sponsor, Manitoba Canola Growers, for sponsoring this morning's refreshment break. Remember to check out the posters during the break. There'll be some of the presenters in the room that can answer some of your questions. And we will see you back here at 10:45.
Questions